label-free immunodiagnostics?
First of all, I'd like to thank the Nano Center for assisting me in supporting setting up the SRIB system that I'll be describing. I also want to start this lecture first and foremost to give you some motivation or explanation as to, as a biologist, what we're actually trying to do. So it's a novel approach towards label-free immunodiagnostics, and therefore, at least for the first half of the lecture, I'll start about talking what do I mean by immunodiagnostics. Is there a...
Use the mouse.
Yeah, no, it's okay. So as a brief description of immunodiagnostics, we're talking about antibody binding towards antigens and targets that we would like to measure in blood. So this is the structure of an antibody, but again, I recognize here that we have a mixed crowd. So let's simplify this a bit. If this is the CPK structure of the antibody with the two binding sites, a more simplistic view is what we see here. Antibodies are proteins of about 150,000 daltons, but they're made up of two heavy chains and two light chains. And another interesting aspect of the antibody molecule is that there is a variable region in which, as you can see, there's a variable light domain and a variable heavy domain. Two such binding sites exist per each antibody, and therefore antibodies are molecules that can cross-link by virtue of their bivalency. Now, a lot of physicists deal a lot with radiation. We all use, for example, remote controls and so on. In the biological world, we're talking about actual binding and physical complementarity between molecules. So what you can see here is a crystalline structure, backbone, of the variable domain of the light chain that I described in the heavy chain. And this is the antigen, that protein which is recognized by the antibody. And as you can see, there is actual physical topography and complementarity, not only in the electrostatic fields and complementarity, but actually physical geography between the two, which create this complementarity, and that's what we're looking at. This is what proteins and antibodies do when they bind each other. Now, another important aspect is that the antigen, which is recognized here by two antibodies, actually contains many sites that antibodies can target. We refer to that site as an epitope. So antigens can contain various epitopes. Antibodies do not recognize the entirety of the protein, but rather specific targets. And as you can see, B-cell epitopes, or the epitopes that are recognized by antibodies, they tend to be discontinuous. And they also are the result of conformational changes and folding of this polypeptide chain, the protein. And so you can see that they can be discontinuous and conformational. They usually contain thereabouts 20 amino acids, whereas proteins usually are in the realm of maybe 200 to 600 amino acids. So this gives you some sense of the size of what antibodies actually recognize. And they normally are comprised of 2 or 3 segments of the protein that are brought together through the folding of the protein such that they create a contiguous surface. The footprint, which is recognized by the antibody, is about 700 angstrom squared. And the important factor here, which I'd like to stress in the concept of immunodiagnostics, is that an antigen contains multiple epitopes. Okay, so that gives you a crash course on what antibodies do and how they bind their antigens. So in reality, when we are subjected to a viral infection, to some sort of immunological insult, as we refer to it, the immune system generates antibodies which are highly, exquisitely specific for the epitopes which are harbored in the antigens of the virus, the bad stuff. And so these antibodies bind the antigen, the virus, so as to knock the virus out and clear it. And that's what antibodies do. Now, of course, once the virus has been cleared, we're left with antibodies in our system. So in fact, monitoring the presence of specific antibodies is a proxy for telling me I've been exposed to a virus, a bacterium, This is the result. Antibodies represent, in fact, the footprint of infection. The virus, the bacteria may be gone, long gone, but the mere fact that we have these antibodies in our serum, measuring those tells us you've been infected by measles, HIV, or whatever. So the first thing that you want to learn, those who don't work with antibodies all day, is how do we normally measure the presence of an antibody? And so I'm sure you've heard of a number of these assays, one of which is the ELISA assay, which basically says the following. We have a 96-well plate, and this plate has a series of wells. And if you take that well, which is in a volume of about 200, 300 microliters in volume, we can immobilize to the surface of the well that antigen which contains a whole bunch of epitopes. And in fact, we can take an entire virus and we can solubilize it, so there may be 10, 20, 30 different antigens, each one containing 10 or 30 different epitopes. You could have hundreds of epitopes immobilized to this plastic well. And then we subsequently take a sample of human blood or animal blood, which contains the antibodies. The antibodies recognize and bind their corresponding epitopes on the antigen. And then we enter another antibody, which was a goat-derived antigen, which is specific for human antibodies. And so under those circumstances, we have a sandwich structure, but the goat antibody actually is linked to an enzyme. So if we introduce the substrate of that enzyme, it will generate a product, which is usually color, and as you can see, you generate colors in your ELISA plate. Well, and this is measured spectroscopically. So this is how we measure antibodies. Now, let's take the case of a virus. There may be, as I said, maybe hundreds of epitopes, and when we do an ELISA test, we can get a whole bunch of colored dots indicating that there were antibodies in these particular systems. Now, the first dot is typically a control. This is where we know we're taking antibodies for that virus. We can see, and we can see the level of the response here. However, we can also have another response, which is similar to the control, and this may be a poor individual that was actually exposed. This is a true positive. However, very often we also encounter a situation where we have false positives. And so immunodiagnostics, the job is to ultimately be able to not only detect the signal, but discriminate between false positives and false negatives. So how do we go about now confirming who is who? So we have yet another test, and the other test is Western blot, and I'm specifically trying to give you some of the terms that people normally use and bounce around. And Western blots basically take the virus, but instead of looking at the gemisch, all of the proteins together, they resolve these proteins electrophoretically into specific bands and then react each antigen separately. So what do you get? In the case of what I showed you previously, the control has all of the viral proteins, and in fact, it has antibodies to all of them. So the pattern we get are all these dark bands which indicate each and every one of the antigens. In the true positive, we'll see that that patient actually mounted an immune response which binds multiple antigens. So by deconvoluting the mixture of all the antigens in one pile and separating them out in space, the fact that we get multiple and distinct different signals gives us the confirmation and the validation that this is a bona fide, genuine, true positive. However, antibodies also have some degree of background, and if there is a fortuitous cross-reactivity in this particular patient, which he has antibodies, but he's only recognizing one band, we would disregard that signal and say that that's a false positive. So in essence, immunodiagnostics is about detecting signals. Western blots are in essence a two-dimensional array of antigens. So we decided to take this one step farther. We said we can strengthen the diagnostic power of our system if we were to deconvolute all the antigens into epitopes. So we would have, in fact, maybe 1,000 bytes of information as opposed to maybe 10. And under such circumstances, maybe we would be able to get a better diagnostic platform. This is what we're trying to do at the Life Science Building. So in fact, we're trying to generate epitope arrays with the sense that this would improve the diagnostic power of immunodiagnostics. So in essence, each marker, we would like it to have 100% sensitivity. 100% sensitivity basically means that all the positives, anybody who is genuinely sick or positive, he will always respond. And this would be 100% sensitivity, 0 false negatives. We'll never miss somebody who is actually infected. And then on the other hand, 100% specificity would mean that we would never have any false positives. That is to say, somebody who's negative never responds. Unfortunately, life is not that simple. And so we never have markers which are 100% positive and 100% or 100% sensitive, 100% specific. So here's where we introduce a new concept in immunodiagnostics, and that we refer to as combinatorial diagnostics. The idea is the following. Let us assume a situation where we have 20 distinct markers, and the 20 distinct markers have very mediocre immunodiagnostic power. They have sensitivity of 45%, meaning, in fact, 55% of the genuine positives are missed. And they have a specificity, meaning 5% of the negatives will show up positive. So this is, of course, intolerable in the industry. You would never be able to use these markers in order to develop a diagnostic kit. So in essence, if we do the distribution of the negatives versus the distribution of the positives with these conditions, we can see that in this particular case, 38% of all negatives would score 2 markers. And on the other hand, we can say 45% of the positives would score between 9 and 11 markers if we had a panel of 20. Now, if we change the number of the markers, as you can see, we can separate the two populations, and we create a window of discrimination, meaning that as we increase the number of markers, we get a resolution and a separation between negatives and positive, even though we're using mediocre markers. Now, we don't always have to use truly bad markers. We can try to improve their specificity and sensitivity as well. So if we go and change the sensitivity, and we use 25 markers, we can show that by increasing the sensitivity of the system and generating markers of improved and even 90% sensitivity, we can get a dramatic separation between the negatives and the positives. So an epitope array appears to be a powerful way of doing diagnosis. Now, of course, the last parameter would be to change the specificity. And as you can see, monitoring from 92% specificity to 98%, again, we can change the behavior of the two populations and generate a situation whereby an array of epitopes would be good. So this is what we're trying to do. We can also see that in a situation, for example, as I showed you previously, 98% specificity, 50% sensitivity, and the window of discrimination is, as you can see, rather large. If we improve the conditions or not, we can get different patterns and try to ascertain what kind of epitopes, what kind of panels would we like to construct in order to get the immunodiagnostic power we're seeking. Okay, so this is the situation. Just to give you a sense that this is not only theory, we can actually do it. If we take a virus like HCV, and HCV has a whole slew of antigens, what we have done is that we synthesized 68 of them and then tested them for the negative, their sensitivity, their specificity. We came up with a panel which ultimately had 12 markers. So this is in the real world. We actually synthesized these things and then asked, do they have any diagnostic power? Here's the distribution of our markers within the antigens of HCV. And here we take bona fide samples from patients. And we ask, if I take the panel and react it in an ELISA test that you already know what that is, then you can see that, for example, HCV1, this particular patient, recognized 9 markers. And we can see that all the positives recognize at least 4. And all the negatives we chose such that even in a panel of something like 30 negatives, none were recognized. So we went for higher parameters in each direction to see if we can get a good diagnostic. Now note here is something curious. If we look at marker 10, it's got an exceptionally high signal for one patient, and in fact, no signal for 2. And the question is why? If it's a good marker, why is there such a discrepancy and such a variation within the population? And so what we decided is we recognize that each virus has its own idiosyncrasies. We have subtypes of HCV, of hepatitis. We have subtype 1, 2, and 3. And it turns out that this particular patient, we would argue, is probably subtype 1. However, if we were to make additional markers, increase our panel, we might have the ability to introduce not only the detection, is he HCV positive, but also serotype. Now, this is important for the clinician because each subtype has its own therapeutic regimen. We use different drugs to treat HCV if it's subtype 1 versus subtype 3. So this has got actual relevance. And in fact, as we increased the panel, we realized that, in fact, HCV4, which previously did not respond to this marker, all of a sudden does. And so we realized that this system not only is effective in diagnosing yes or no the disease, but also serotyping. So we were pleased with this. Now we have a problem. I'm switching hats, and I will try to give some of the background as far as the physics as to what we're trying to actually do. So the objective is the following. We want to take a silicon wafer, and we want to spot it with an array of between tens, 20s, maybe hundreds or thousands of epitope markers, so that we can devote maybe 10 or 100 markers per each pathogen, and maybe actually have a chip that will allow from a single drop of blood the diagnosis of 20 or 30 different viruses. And so this is the strategic goal. But now you have to say, in the real world, we can't use ELISA plates, because each ELISA plate would require, if we're going to take a baby blood sample, we would have to drain them of whatever blood they have in order to do 1,000 swells. This would not pass. So what we have to do is try to reduce concentrations and put it all onto one chip. And for this, we decided to go to optical reflectance and interference. So at Boston University, on my sabbatical, I worked with the Nanophotonic Center there, and what we developed is the following machine, which is currently in the life science building in our lab. And again, it's my opportunity to thank the Nano Center for giving me some of the support to purchase it. So this is SRIB. This is actually the picture in the real world. But so as to give you a description of what we're actually looking at, this is a description of the machine. As you can see, we have a light pipe which is going in. We have a tunable laser, which is allowing us to sweep from 765 to 781 nanometers. And the light pipe introduces the light from here. Here we have a beam splitter. The light is reflected from the sample, back up and reflected into the camera. And then we quantify per pixel. Each feature constitutes approximately 100 pixels. And so on a chip of about 1.5 centimeters squared, we can easily spot somewhere between thousands and maybe 10,000 different spots. So this would be the idea. Okay, some of the physics as to what we're actually trying to do. We have a standard silicon wafer in which we have thermally grown silicon oxide. This, therefore, would be the interface, and that is our buried reference surface in which we're going to get reflectance there. So if we have a beam, part of the light is going to be reflected at the interface silicon oxide, air, or water, and the other is going to penetrate the silicon oxide and be reflected by the reference interface. And the thickness of the silicon oxide is about 10 to 20 microns, in this case 15. Okay, now what does this mean? So what we're doing is we're going to be measuring the intensity of light per pixel. And so if we look at the beam or the wave, and here I want to thank Adi for giving the background. As you can see, we have two reflections. One is delayed. The black is delayed with respect to the red. And the extent of delay is obviously immediately relevant or related to the distance and the thickness of the silicon oxide surface. It is delayed by that amount of time that it has to penetrate and be reflected and back to the surface. So, of course, one does not observe two beams, but rather observes a single beam, which is the product of, in this particular case, constructive interference. And so here we can measure the intensity. We know the light wavelength. We are measuring the intensity per spot, and from there I can actually derive this thickness. Now, if on the other hand we spot onto the surface of the silicon oxide a protein sample, so we're going to get now a series of reflections. One reflection is going to be reflected from the surface of the protein itself. And so under those circumstances, we're going to add to the 15 microns another thickness, which is in the realm of a number of nanometers. And so again, here, if we follow the beam, the red beam starts first, but then delayed not only for this, but also for the distance here, we find the black beam. And of course, again, we don't see the two beams, but rather we see the product of the interference. In this particular case, and I took it just to be a little more provocative, we can see that in fact this interference is total destructive. Now, to some of us this might be a bit paradoxical. We've added height, and yet in view of the fact that we've added height, we've actually eliminated and canceled all light. So the reality is that you have to understand that this is also oscillating and it has a periodicity to that. And therefore, under the circumstances, what you want to realize is that if we take the black beam and delay it each time as a function of the height that we're adding, we're going to see an oscillation of the intensities that we would meet or measure for each pixel. And so again, as this is being delayed, we see an increase in signal, but once it follows and passes the peak, then it goes down to the lowest level, and then back up as we approach the constructive, and ultimately down to maximal destructive interference. And so through that, in essence, what we have is an oscillation in the intensity per each pixel that we're measuring, and that is going to give us the information and the ability to calculate what is the added height at each feature that we've actually immobilized to the surface of the chip. Now, there are two parameters that are going to affect the intensity. One is the height, but the other is directly also the wavelength that we use, and therefore we're using a tunable laser and measuring at a number of different wavelengths. As you can see here, let's take three different heights, 0, that is the baseline level of the silicon oxide, and then 10 nanometers above or 20 nanometers above. As we go through the different wavelengths, we're going to get different intensities. And for this particular wavelength, 774, as just an example, you can see that the lowest level, the ground state level, is actually going to give a signal which is the highest. It's the lightest. Whereas the highest level is giving the darkest. However, if we're going to go to another wavelength, going to the same level, as you can see here, it flips. So by combining both parameters, the height and the wavelength, or actually measuring the intensity and knowing the wavelength, we are able to derive the height at each feature, which allows us to visualize, if you will, binding. Now again, I want to go to the practical and end the lecture with some raw data. Here what we've done is the following. We've taken one of the wafers and we've spotted it with 200 samples. So we're looking at imaging all 200 at once. The 200 samples are just repeated rows of 4 different proteins. The first protein is protein G. Protein G is a bacterial protein, and it has the unique characteristic that it binds immunoglobulin. So it's just going to arbitrarily bind antibody. So it'll give us a sense that we can spot, and we expect as we flow in antibody to see binding at those spots. Now, the second series of spots, which we screwed up a bit in our spotting, so you can see it's a little dirty, is rabbit immunoglobulin. It's simply antibody derived from the serum of a rabbit. And then what we did is we took 2 proteins which are typical of serum, one from humans, so it's human serum albumin, HSA, and the other one is bovine serum albumin, 2 very common proteins used in biological labs. Once we spotted all of the chip, we then reacted the chip first with goat anti-mouse immunoglobulin. So this is an antibody derived from a goat, Schadgadya, and the antibody derived from the goat will be bound by the protein G. And so we should see specific binding at those features. Now there's another thing. I want to share with you something about biology. Goats don't always discriminate between mice and rabbits. So under the circumstances, the immune system of the goat is going to mount antibodies to the rabbit. However, there will be, or to the mouse in this case, but there will be some degree of cross-reactivity, some limited binding of the goat anti-mouse to the rabbit immunoglobulin. This perplexed my colleagues, engineers and physicists in the States, but in reality, it actually shows us the sensitivity of the system because we can also see the marginal response. And finally, we will react with rabbit anti-human serum albumin, which binds to the human protein as well as to bovine. Let me show you what this looks like. This is a collection of 16 signals for each one of the spots, that is each one of the types. And as you can see, as we flow in, well, let me just show you the results. Here we've put it onto a time lapse, and what we'll be seeing is flowing fluid, looking at real-time binding as we have on the level of the chip. I didn't expect to have to try to find my... Okay, so as you can see, we're flowing fluid. The goat anti-mouse comes in, and you see the development of the spots, which is this series rising. After a few seconds, we can see binding to the goat anti-rabbit IgG, but no binding, as you can see here, to the human serum albumin or BSA. Once we introduce the rabbit anti-albumin, we see the signals first and foremost for the human and then to the bovine. If we then subsequently introduce HCl, we acidify. This causes dissociation of the complex, and we lose our signal. So let me just summarize under the circumstances. Half a minute. Basically what I wanted to do is share with you, first of all, a new concept in immunodiagnostics. And therefore, what we are suggesting is to increase the power of diagnostics by making epitope arrays. It is the same logic as going from an ELISA to a Western. We want to go one step further. But epitope arrays are going to require some sort of a system, a measuring system, which will allow us to look at tens and preferably thousands of spots cheaply, in small volume, and with some degree of efficiency. This is why we've introduced the SRIB. I hope I've been able to convince you that it is both interesting and hopefully useful. And with that, I'll just thank you. Basically, Dror Simantov, the PhD student, started with the combinatorial diagnostics, and Aryeh Rifkin is the student who is now introducing and working with the SRIB system. Rani Turkaspe and Romy provided the materials for the HCV analyses, and my colleagues at Boston University, Bennett Goldberg and Selim Unalu, and Emre and David Bergstein, who has since started a company called Zorray Technologies, in which he is hoping to be able to market the system that I've just described. Thank you very much.
We have time for 2 or 3 questions. In your measurement, you assume that you have the same mathematic index for all the
Yeah. No, actually, in reality is this. In such an audience, obviously, I have to not go into all the finalities of exactly what we're doing. We don't actually refer to nanometer heights, but rather optical thickness. An optical thickness is a compound concept or factor which takes into account the refractive index of the protein. But in practical sense, we are assuming that proteins in general have a refractive index very similar to the silicon oxide. So without overcomplicating the system, this seems to work. We are calculating what is perceived as optical thickness using MATLAB algorithms and so on. So this is a visualization of what we're actually calculating. So you're right. The refractive index obviously contributes much. Yeah?
Since it's Thomas' question, I'll ask the simple one. In this movie that you showed,
Yeah.
this was for one laser frequency, or you're also scanning the laser frequency that you described?
Okay, so this is actually a compilation of a number of laser frequencies and calculation of all. In reality, which again, I did not tell the complete truth, what we actually do is we spot. We take a measurement, but then, and again, both in ELISA and in this SRIB analysis, we have to block all other surfaces so they don't non-specifically absorb our antibodies or reagents that we're incubating. Therefore, if you were to look at the surface, you would just see 0 because it all goes to the same level. We take a picture of that, and that is our background, and we subtract that from each and every frame over time over all of the frequencies. and then subsequently put it all together, calculate, and generate the image. Okay?
Any more questions?
It's interesting, biologists and everybody's got their own angle. I don't know. No. In reality, what we have is this: we can spot certainly within the less than maybe around 100 or some picograms, and we can detect around 100 nanograms in solution. But I would like to hedge a bit because I'm not totally confident that I can actually quantify in our spotting system what we're actually putting down. When you visualize initially, you're going to see a lot of salt, and we wash it away, we don't see anything, unless we do the SRIB analysis. So it's very preliminary. It's in the lab now. It's going to take us a number of months to get it up and running, and then if people want to try it, sure,